Interest Group, and we are broadcasting live in Stanford University. Okay, I'm C.H. Ting, and this morning the topic is pattern search in non-coding RNA. Actually, I had the newer title, which is code in non-coding RNA. So I didn't. I thought I update the slide, but not everything. Okay. Since that I would talk this morning, this is a summary. I would talk a bit about long non-coding RNA, and there's lots of data in long non-coding RNA on the web. But the data sets are huge. So I would do some pre-processing on the long non-coding RNA databases. And once the data is formatted correctly and pre-processed, I'll do exhaustive search on all the patterns that's useful, meaningful in the long non-coding RNA databases. And then I will extract what I call the pearls and necklaces. These are the meaningful thing, and these are code in long non-coding RNA. And I would talk about how to interpret, how to make sense of this code, the data, the pearls and necklaces. So I would follow what people termed as the central dogma. This is the basic principle in microbiology, which states that information are copied from the DNA to RNA and then to build proteins. So this is a central dogma. But this is not sufficient to explain everything in microbiology. So I would propose a computer model for all cells. And this I call cells are just like computer. It's very much like computers. So how this computer operates, and I will propose what I call the general dogma. And this explains almost everything that we know about the cell, about the DNA, about RNA, and about protein. So this is the schedule for the morning. And so we got lots of information in genomic data sets. So the genomic information, but currently most of our understanding of the genomic information are coding DNAs. And the coding DNAs are generally called genes in the DNA. And these are segments of DNA which encode proteins. Okay. And this generally they are called coding DNA because they have code which eventually translate into protein. And intermediate step in making the protein is to copy the information from DNA into coding RNA. And the coding RNA mostly are what we call the messenger RNAs. And these messenger RNAs are later translated into protein. So the first step from DNA to RNA is called transcription. It transcribes the protein code from DNA to RNA. And the code in RNA are translated into a protein, into many proteins. So that's most of our understanding of the DNA. We know coding DNA, which builds protein. But in the human genome, only 1.5% of the DNA are coding DNA. So there's 98.5% of DNA which not used to code the DNA. And earlier, microbiologists they are very proud group of people, and they thought they know everything. So everything they know are the coding DNA, and the parts which are not coding DNA, they call junk DNA. Okay, and they say, well, it's useless. Okay, so they are junk, but the junk is different from garbage. Okay, garbage are things that you throw into the garbage can to be recycled or just get rid of. But junk are something that you save in your garage, okay? And you say, oh, maybe sometime I'll use it for something. So that's junk. But now, much later, people realize the junk DNA are really not junk. There are information in it, and there are important information in it, and which we just don't understand, because the idea is that the purpose of DNA is to make protein, and things that does not support protein production are useless. So we have this idea that information are copied from DNA to RNA, and from RNA move on to the protein. So the part of the DNA which is not used for protein are called non-coding DNA. And there are lots of non-coding DNA are transcribed into RNA. And people know that these RNAs are not used to produce protein. And now what are they used for? So they call these RNA, which are not used to produce protein, long non-coding RNA. And these long non-coding RNA are RNA molecules which has 200 or more base pairs, and which do not encode proteins, or which do not serve known or useful functions. So long non-coding RNA, there are lots of different DNAs. Long non-coding RNA are arbitrarily defined as RNAs which are bigger than 200 base pairs. So in the long non-coding RNA, they exclude messenger RNA. So messenger RNA, we know they are used to produce proteins. And tRNA and rRNA, these are RNAs which are used by the ribosome. And ribosome is the machinery which translates messenger RNA to protein. And then there are lots of small RNAs which are called microRNA, siRNA, snRNA, and many others. And mostly they don't know what they are doing. Only a very few things people have identified them and found the kind of control the expression of protein in one way or the other. So there are lots of RNA. There are lots of long non-coding RNA, and there are lots of smaller RNAs. So all these things needs explanation we have to understand all these things, what they are doing. So the information in DNA are first transcribed into RNA. And why are they translated? Okay, so we know if they are messenger RNA, they are used to express, used to translate into protein. But why are all lots of other RNAs? What are they doing? So long non-coding RNA has to contain useful information because they are translated and then move out from the nucleus to the body of the cell, and do so. We don't know what they are doing, but they are there, and cells are known to be efficient in utilizing its resources. So it is hard to imagine that long non-coding RNAs are transcribed for no good reasons. And we know from many all the microbiology reactions and cycles, we know that the cell operates very efficiently. It uses its resources very efficiently, and it try to generate the energy from food very efficiently. So and it reproduces. So it duplicates DNA and make another copy of itself. And all these things are very efficient. So it is hard to imagine that cells are transcribing long non-coding RNAs for no good reason. So my thinking is that long non-coding RNA must contain information highly condensed and actually used by a cell. Now, what kind of information do we expect in long non-coding RNA? How do we search for information stored in the long non-coding RNA? So my mistake last year was looking for information in DNA. And I was kind of disappointed in not finding any useful thing there. But this year I started looking at the RNA because DNA, the information in DNA is spread out, okay? And only the useful thing are copied and edited in the RNA. So, I would concentrate my search in the human long non-coding RNA, because the human long non-coding RNA is the biggest collections of long non-coding RNA. And I thought that I need a complete set of long non-coding RNA to do meaningful analysis. So here are 5 databases with long non-coding RNA for human. Okay. So the first one is in the genome project, and they located some non-coding RNA, and it's only about 64 megabytes of size. But there are bigger collections, long non-coding pedia, which has almost 200 million bytes. And the non-code, this is another project, which has almost 300 megabytes. And this one, cDNA, is very interesting because they very early in the genome project, people had found that there are lots of RNAs in the brain tissue, Okay, so they extract lots of RNAs from the brain tissue and start to analyze them. And the best way to analyze it is to reverse transcript. They translate the RNA back to DNA because DNA is much more stable and much easier to analyze. So they translate this RNA back into DNA and call them cDNA. So I think these cDNAs are actually RNA. So I try to include this in my analysis. So, and then later on I found a much bigger long non-coding RNA, which is almost, which is 400 megabytes long. So I have lots of data that's published and available to be downloaded. So I download all these things and start to work on them. So the information, what do we know? What's information? Okay. In a big data set of anything, images, pictures, characters, text, in any big collection of data in any form, information are repeated patterns. So patterns which are not repeated generally does not have meaning. If you want to search for meaning, you look for repeated pattern. And when the pattern is repeated, and if they are repeated a lot of times, they have importance, they have significance, and they have meaning. So for the RNA, long non-coding RNA, I arbitrarily select 20 base pair patterns. I say, well, if I'm looking for information, how do I start? So to start with, I'll just look for base pairs patterns. There are at least 20 base pair long. So I assume that the information will be 20 base pair or longer. And the goal is to find all repeated pattern with 20 base pair or more in all the long non-coding RNA databases. Okay? So we are dealing with huge databases, up to 400 megabytes long. And the long non-coding RNA databases are first formatted in records. So to facilitate analysis, because all the RNA databases are published in kind of in some standard format, and they are just mostly called FASTA or FA format. And this format is that for every gene, for every RNA, or every chromosome, they would publish the data in a very simple format. And for every gene, the first line is a name or comments, okay, and which has a greater sign in front of it. And follow that line, the data comes line by line. And each line generally are 60 characters long, with a carriage return and line feed in the back. So so this data are just a big text file, and they are lines. And the first line is a comment, a name, and then followed by many lines of actual A, G, C, T text. So I reformatted this thing into the records that I can handle conveniently. So every gene, every separate entity in the long non-coding RNA database, I would build records. So the records have two fields. First field is name. I just extract a short name, a character, a character sequence that can identify this RNA. So the first field is a text field with a name, and I preserve the greater sign, so that later I can see things much clearly with the greater sign, which separates itself from the text. And the next field is a variable text field, which contains all the base pair, and in one text file, however long that RNA is. And I look at the data. The longest one is 32 kilobytes. Okay, it can be up to 32 kilobytes. So all the genes, all the RNAs are lined up in my data file in a tab separated fields of records. And so I first formatted in this field so that I can, for one thing, okay. And sorry for everybody. I'm doing all the processing, all the calculation, and all the analysis in Python. Okay. And when I format everything in this format, it's very convenient to display the data in Excel. And Excel has also lots of capability in doing the text analysis, and especially in formatting your data. So, earlier I was doing lots of everything in forth, but I found that once the most difficult task is to extract all the 20 base pair repeated pattern. So this I had to do it in forth. But later on I found that I can improve the algorithm such that Python can do it successfully and not similar to forth. So once I pass that threshold, I just move everything into Python, because Python is really very convenient in handling text, doing search, rearrangement, and the formatting, and these things are very easy to do in Python. And the code becomes very small. Especially Python is very good in file. So I can open up lots of files and do things in each different files. And when I collect the result, I just write them out into files. So this reading things into file, do a little bit of processing, write it out to a file for later processing. So, Python is very good. I think I can do everything in forth, but it's becoming quite laborious because even open up a file in forth, there's a long it's a long Windows call, and in Python you just say open. Okay. So, long non-coding RNA databases in FASTA format are converted to data file with records separated into 3 fields. So I have name field, or I have a length field, and then data field. So originally I just separate into name field and data field, but later I found having the length is very, very important. So I add a length field for all the records. And when the entire file is converted into this format, I can read the entire file into Excel and tell Excel to delete all the duplicates. Okay, so that was very, very nice. And if I found that there are lots of duplicates, so all the data files, they collect the data. These guys collect data from everywhere, and just collect them, and not actually having good method in shifting them out. So the first thing I did is to throw away all the duplicates. And once throw away duplicates, very easy to do. It's just one button in Excel. But I can do a much better, because there are RNA molecules as they recorded in the databases. They might have duplicates, and they might have longer, shorter RNA replicate inside bigger RNA. Okay, so these cases I need to sort them out. So what I can do, this has to, I think the Excel is not good enough to do that. So it has to be done in Python script. So records are first sorted according to its length. So the shorter one comes first, and the longer ones come later. So I pick up every RNA and search downward to see if any of the RNA is embedded in a bigger RNA. So if it is, I just throw it away. The shorter one, yeah, because the information is in the bigger one, so I can afford to throw away the shorter one if they are embedded. Okay, and so this is very, very nice. And going through this process, I can throw away almost 10% of the RNA in databases. So the records are then resorted in ascending names. So I have the data kind of aligned to how they arrange things in the database. So now I have unique, or I throw away all the duplicates, and I also throw away the duplicates which might be embedded in other RNAs. So once I have that database cleaned up a little bit, then I will start do the search. So the first step to do the search is to go through the entire database and collect all the data into a single flat data file for analysis. And I just record the pointers to the beginning of every RNA to a separate index file. So I have a data file and an index file. The data file contains all the data in a flat file. And the index file has 3 fields. It has a pointer, which points to the location where the data of RNA is stored in the data file. And a name field, I keep the name and the length field. So the index file has the pointer, so I know where things are, and the length, so I know how long this RNA is. And also I keep the name for later sorting and identification. So once I have separated this thing, and when doing search, I will do the search in a flat data file. Okay, and so there are problems that you have two RNAs right next to each other. So you might have interference between different adjacent RNA, but I think that's not a big problem. And if I have to do the search in individual files, I can do it, but the things are getting more complicated. It's much easier if I have all the data collected in a flat data file and pick up all the repeated pattern, and then later on figure out which pattern belongs to which RNA. So all data are stored in a flat data file. And now I go into look for repeated 20 base pair patterns. And these are called repeats. So I just locate all the 20-bit patterns which are repeated. If they're not repeated, I think they might not mean anything. If they are repeated anywhere in the data file, I'll make a record of it. Okay. And so, so I have collected huge amount of repeats. What do I do with the repeats? Repeats are sorted according to the pointer, according to their addresses. And I found that lots of repeats occurred consecutively. So you move the pointer one base pair every time. And there's lots of things which just repeat, lots of repeats are consecutive. Okay. And the consecutive repeats are packed into pairs. Okay. If I have sequence of repeats which are consecutive, they belong to the same thing. Okay. And they are compact into variable length data. And these data are called pairs. And the cluster of pairs are necklaces. So I'm expecting lots of these repeat. Now pairs are repeated patterns, but they are not limited to 20 base pair. They can be longer. And some of the pairs are just as long as the very long non-coding RNA. They can be as long as 32,000 base. Yes.
The cluster of repeat?
Yeah. The cluster is the pairs. You know, repeats. There's lots of repeats which are consecutive. Okay. These repeats are packed into pairs. Okay. And the pairs, if you look into the data, pairs are separated repeated patterns. And these patterns are not are 20 base pair or longer. Sometimes it's very long. Okay. So these are pairs, and the pairs, lots of short pairs occurred in clusters. Okay, and these are the things I'm looking for, because pairs are information. They are repeated patterns. Okay, they repeat in the database somewhere. Okay, and when the pairs occurred in clusters, and they have really very useful information. They have very useful meanings. So what I'm looking for is from the repeats, try to locate pairs. And in the database, I'm looking for clusters of these pairs. But if you have been, I think you guys have all been here for a long time. You know what I'm at. These clusters are fourth programs, okay? And that's what I want to find and to support my assertion that genomes are programmed in fourth. Okay, pairs are repeated pattern, 20 base pair or more. And there are lots of them, and they are of variable length. And they are arbitrarily classified as long pairs, which are 200 base pairs or longer, and short pairs are 50 to 199. And anything smaller than 50 base pairs, I call them micro pairs. So the nomination, I think I use the pairs and necklaces deliberately to be different from the microbiology terminology. So I don't want to call them RNA or DNA or this well-known terminology, and I call them pairs. And these pairs are information I identified in long non-coding RNA databases. So we have very long pairs, and this is just like long non-coding RNA, which are defined as RNA molecules 200 bases or longer. And then microRNA is microbiology found lots of microRNA, and they are 18 to 22 bases long. So if they are in this range, it's almost like microRNA. Then short pairs, they are most likely functional. And we have found lots of, for example, the transfer RNA, which assists the building of the protein molecules. And these transfer RNAs are about 70 to 150 base pair long. And this serve useful information. So they are most likely in this range. So I can kind of try to understand, classify them, and try to understand them in that way. Long pairs are repeated patterns, 200 base pair or more. So, so if you look at the data after the search, after the pairs, there are lots of very long pairs. And they are thousands, they can be thousands of base pair long. But one reason why there are so many is that if in the database, for example, in the database I have 2 RNA, and if the 2 RNA they have say 1,000 base, which are the same, which may not be at the same location, okay, but they occurred. A stretch of 1,000 bases occurred in 2 different RNA, and this will cause cause the recording or the recording of 2 1,000 base pair pairs in my analysis. And so they are just simply repeats in the RNA database. But when they if we accidentally have these two RNAs as two separate RNAs in the database, I will just have these two 1,000 base pair pairs. And the internal structure of this 1,000 stretch are masked. So I don't know if there are microRNAs, micro pairs in this 1,000 base stretch. So if the data has repeats in the database, and it tends to mask off its internal structure. So that's the reason why I have to throw away all the repeats, and I spend lots of time looking for shorter RNA in longer RNA. But just looking for shorter RNA in longer RNA doesn't guarantee that you catch all these repeats. So I need to look at them much more closely and try to eliminate as many of them as possible. So these are the repeats. They are redundancy in the long non-coding RNA database. But another possibility is that these long stretches of RNA are actually useful, okay? And so if I can eliminate all the redundancies in the pairs, and finally I should have one set of RNA which are functional, okay? So this is okay. Clusters of micropairs within each long non-coding RNA can now be identified as necklaces. So you see, I have a huge flat data file, and I have an index file which allows me to identify each and every individual RNA. So after I get all the pairs out, I would sort all the pairs according to their pointer. And then I re-merge the index file with the pair file. And now you can see I insert the header of each RNA. And the headers will separate out each individual RNA molecule. Okay? And then I'm looking for repeats, the clusters of pairs within each and every long non-coding RNA. So this is the way how you can identify. And if the cluster, if they cross the boundary of two RNA, that doesn't mean anything, right? So the clusters has to be pairs within one long non-coding RNA. So, and only the clusters within one long non-coding RNA, and these can be identified as necklaces, okay? But I found lots of necklaces, okay? There's a huge amount of pairs, and there are lots of necklaces. They occurred in many, many different long non-coding RNAs. Okay, there's lots of long pairs persist in the pairs file. So they can still be repeats, there's still be duplicates of RNA stretches, but they could be they could have function. So it's important to remove redundancy in the data file. And I thought in the long non-coding RNA database, I talk about how to remove redundancy there. So I can delete all the repeats, all the duplicates, and then I would search for a duplication of shorter RNA in longer RNA. So I had the 2 steps to reduce redundancy at the database level. And once I do the per analysis, then I still see lots of long, very long pairs. And so the redundancy, are not completely removed. Okay, so I go into the per file, and then try to identify redundancies in the per file. Okay, so this is the another level of redundancy removal. So similar to what I did in the long non-coding RNA data file, I look at all these long pairs and sort all them according to their length, and then look for shorter pairs in the long pairs. And I think if a shorter pair occurred in a longer pair, that might be redundancy. So from that, I would go back to look for these long pairs in the original data file. If I can see these long pairs in 2 different RNA, I would try to remove the shorter one. Okay, so this is from the per file. If there are redundancies in the per file, I would move the record the redundancy and go back to the RNA file. Try to remove the redundant. Try to remove the RNAs which has redundant pairs in them. But even with that, there's still lots of redundancy there. So you can see here, these are the original data file, okay? And I can identify, there are RNAs in, lots of RNAs as annotated in the data file. So if I do all the redundancy removal, what I end up was kind of removed about 1/3 of the RNA, which I can claim that they have redundancy in them. And so the RNA, there are lots of RNAs being removed. But here you see the RNA was, there's still lots of RNA. And these are the pairs remaining in the per file. So these are the data I have. These are my analysis. So there are enormously large number of micro pairs and short pairs. Okay, and there are lots of micro pairs and short pairs which form necklaces. Okay, and pairs and necklaces are coded in the long non-coding RNA. These are the meaningful things. Okay. So, the title of my presentation today is Code in Long Non-Coding RNA. Pairs are code. Okay, they are repeated information, and because they are repeated, they have meaning. If they form clusters, they have more meaning. Okay? And so if we try to connect the data analysis, the pairs and the necklace to the actual RNA word, so micropairs are likely microRNAs. And they are, I think I found lots of them. Their length is between 20 to 50. But micropairs, so far we found the microbiologists found lots of them with the length 18 to 22. Okay, so I think I have the information in the in micropairs, and these micropairs may be causing the microRNAs. Okay, short pairs are likely functional RNA, and long pairs are duplicates, or some of these very long ones might be functional. We found a few RNA which are actually functional. Okay, especially what they call the ribosome RNA. These are RNA molecules which builds a ribosome. Okay, and the ribosome is a basic machinery which translates messenger RNA into protein. So and there are lots of transfer RNAs. These transfer RNAs are transferring transferring the amino acids into the ribosome to build protein. And these are RNA molecules which actually have function. Okay, so I think there are lots of more RNAs which may have function, but we have people have not been able to identify them, because the protein research has been carried on for 60 years. Okay, but RNA research was done only quite recently, because the tools are not mature enough. So here I just want to show you the beginning of all these profiles. So for the GRCH38 cDNA, you see lots of long pairs. So the first one is 1032 base pair long, and the next one is 2064. Okay, but you can see they are shorter pairs, and they form clusters. Okay, and I think these are.
So that 1, 2, 3, 1, 2, 3, 4, 5, the sixth line down. That long one that goes all the way to the edge of the screen. That continues on.
Oh, yeah, right, yeah. This is a very nice feature of Excel. You look at it. It just, if there's nothing in the in subsequent cells, it just show all the data from this one cell. So there are only 4 fields. Okay, this is the pointer field. Okay, and this is the distance field from one to the next, and this is the length of the actual pair. Okay, so this is a very, very nice way to visualize what we have in the pair file. Okay, so this is from the cDNA file, and this is from the non-coding RNA in the Human Genome Project. Okay, you can see, some pairs here, some clusters here, but there are lots of long things. And I believe that these long stretches still contains pairs and necklaces, but because these they have duplicate long stretches, so information in them are masked. Okay, so if I can further remove the redundancies in this here, I can reveal a lot more information from this very long non-coding, very long pairs. So this is another database, and another database, another one. They're basically showing you the same thing. I only show you the beginning of each file, but if you look down, you can find all kinds of things. Okay, so pearls and necklaces. So I expect that the pearls in various long non-coding RNA databases will be highly correlated, but they are not. Okay, so I try to get all the information I can collect and analyze them. If they are all coming from the same human and they are all recorded with the same technology, each database should have correlated. So there's only so many RNAs from from a single human being, right? If they are all there, database should be correlated, but they are not. Okay, and I think these long non-coding RNA databases are incomplete, or they are collected wrong. Okay, in themselves. I wish microbiologists would do a better job in providing a better collection, so we can see more clearly how, at least in human being, what kind of information we can collect. Any comments?
There's a woman out there that they've collected her DNA and they've spread it all over the world. She's like the standard DNA. Does it all appear the same in all of these various collections? I'm amazed that these collections, are so different, have so many different things in them.
Yeah. We don't know. Yeah, they collected, they are standard, the Genome Project, they select a few people and do the
individual real people.
The data are from real people, but these databases are probably not. Okay, and I found that the later two big databases, and I was looking. Well, I have to look at the source, how they are collected, and the two Genome Project are collected in England, associated with Genome Project, and one database, the Linkpedia, is collected in Holland, and then the later two big ones, they are in Beijing. And what these people do, they just go to basically they do not do the research, and they just go to the literature and do the literature search and find sequences which were published, and they just collect them together. So I think these are fake research, but they are meaningful to a certain extent, but after doing this kind of analysis, you will see that if they are not correlated, they are probably not true. Okay? But I don't know. I'm not doing the actual work. I'm doing the same thing, right? Just copy data from the web and try to do my analysis. So I have I'm not qualified to really criticize them. Not doing the same thing. So the next, I think I would just go quickly. This, yes, 5 minutes.
Okay.
So I have what I propose as the general dogma, and this is actually from the central dogma. Crick, the the first guy who, the inventor of DNA. And he, in 1958, he proposed this central dogma. And it's pretty much adapted by all the biologists. And that claims that the information in DNA is copied into RNA, and the information in RNA is used to produce protein. So this is the central dogma, and which guides the research development in microbiology for 60 years. Okay. But this central dogma is very limited. It does not explain the huge amount of non-coding RNA. So in human, people understand that according to this central dogma, they can explain only 1.5% of the DNA. So what about the junk DNA? What about the 98.5%? It does not have anything. It doesn't say anything about the non-coding DNA. And it does not say anything about the huge number of long non-coding RNA and microRNA. Okay. It does not explain, it does not have a good model of how cell operates. It just explains how protein is produced. Okay? So the central dogma is very important because we see that in the genome there are information, and this information is copied and then translated into protein in a very precise fashion. Okay, so this is the basis of microbiology for the last 100 years. Okay, so I'm proposing a better dogma which explains everything. Okay, so I propose that microRNA is the key of the cell computer. MicroRNA causes transcription of an RNA. So the microRNA, its function is go to the DNA and find out the code that's in there and transcribe it into an RNA. So there are two kinds of RNA. Functional RNA, which performs a assigned function. Some of them is to make proteins. Okay, but we have already know lots of RNA which are not associated with protein production, but they have real function in cells. Okay, and there are lots of functional RNA and informational RNA. These informational RNA, they will release clusters of microRNA. Okay, and these microRNA go back to the cell or to the DNA and causes release of RNA. And this RNA, if it happens to be informational RNA, it will release another cluster of microRNA, and so forth, and so on, and so on. And this explains the complexity of the cell function. And it also accounts for the precision of cell function. Okay, because this recursive release of microRNA, these are programmed, and this accounts for the function of the function. And another very important thing is the microRNA can be released in out of a cell and go into other cells, which would coordinate, which would cause or coordinate multicellular functions. So this is what we have to, it has to explain how we operate. We have billions of cells in a body. And the billions of cells, their functions are coordinated. How can they be coordinated? Okay, and this, I think this is the important mechanism. If the RNA can be released from a cell and go into other cells and causes other cells to function, to perform certain functions. And this would account for the multicellular organism. Okay, so I think 2 more minutes would do. And the general dogma, if we draw it graphically. So we have DNA, and the microRNA goes into the DNA to transcribe RNA. And there are 2 kinds of RNAs. Functional RNA some of the functional RNA would make protein. And there are lots of other RNA which will do things we don't understand yet, but they have, some of them we already understand, but lots of them we don't. But I assert that there are lots of functional RNA which will do specific things, just like proteins would do. And informational RNA, which they just contain clusters of microRNA. And they would release microRNA, and this microRNA will go back and make another set of RNA, and so on and so forth. Okay, so this is what a cell, the mechanism of a cell computer. Each cell behaves like a computer. All code are stored in DNA, and useful information are transcribed and edited in RNA. So this is like a computer. The DNA is a disk drive. Okay, the information in this drive is copied into RAM, and sometimes get edited, selected, and they get executed. So the cell is doing exactly the same thing. Useful code are transcribed and edited in RNA, and functional RNA perform specific functions, and informational RNA release the cluster of microRNA. And this is the. So living cell is a Turing machine, and microRNA is its basic instruction set. Okay, and it has a big primitive microRNA which causes transcription of functional RNA, and it has all the other microRNA would transcribe informational RNA, and informational RNA will release clusters of microRNA, and then this goes on. And, so the cell computer, each microRNA causes transcription of RNA, functional RNA perform functions, informational RNA produce a list of microRNA. And recursively transcribing functional RNA through microRNA, DNA, informational RNA, microRNA loop may account for the complexity of a living cell and its precise control. Okay, and well, you know, it's just like a fourth computer. Fourth, we have demonstrated and proved that recursively processing nested lists can solve any computable problem. So necklaces as cluster of pairs. So this is the evidence I want to find to prove, to show that the general dogma is a plausible mechanism. So a dogma is a belief system, okay? It's supported by insufficient evidence. So we don't have everything lined up to prove it yet, but it is open to improvement. It's just like central dogma when it was proposed in 1958. People really don't know how things work, and people say, well, information can be in DNA, information can be in protein, and the protein information in protein might be, you know, by evolution, go back and change the DNA. the information can be used to change the original information in the DNA. But the dogma, central dogma says no, information flows only from DNA to RNA and protein. Information does not go from protein back to RNA or back to DNA. And this was, at the time, nobody has any proof of this. But the Crick was very inventive in seeing that information has to flow that way. And all the later experiments proved his point. So central dogma was accepted by everybody. So the general dogma may be a useful model for the next 3 generations. Of course, there's lots of things that we don't know. I think I'm not a microbiologist. I'm only a force programmer, right? And so lots of things I cannot do. So I have to prove. There are lots of things has to be proven. Okay, micropers, microRNA. Okay, does microRNA actually cause transcription of RNA? There's no evidence that microRNA can cause the transcription of an RNA. But I think the microRNA has enough information to make to do a very specific function, okay, and which is caused to transcription of an RNA. And this is not that's you know, in the genome research they do the what they call the they copy the DNA. I forgot the term, but they copy you can copy lots of DNA. You can repeat the copy many times, but the copying depending on primer. You have to have primer. The primer is about 20 base long. You have this primer, then the DNA replication can start from this location and continue to the point where there's the same primer in the stretch. So I think there's a little experimental evidence that microRNA could cause the transcription of an RNA. Then how do long non-coding RNA release microRNA? Don't know. We found I can only show you that there are clusters of microRNA, micropers in long non-coding RNA, but I don't know how this cluster are processed eventually to cause the release of the microRNA. So this needs experiments to prove. And I found lots of long pairs. Are they redundant, or do they have function? Okay, could some long pairs be functional RNA? So all these things need to be looked at very carefully in experiments. And the current long non-coding RNA and microRNA databases are not complete. Okay, but even with this limited amount of information, I can show you there are clusters of pairs, and hence it is possible that these pairs would cause the release of a whole set of microRNA. Long non-coding RNA and microRNA are very elusive. Okay, people who study them knows that they are very small amount of them. Even one molecule could cause lots of trouble. Yeah, this is the last slide. Okay. They function, the microRNA and the long non-coding RNA function at very low concentration. So it is possible that you cannot even detect them. And cancer database is naturally not complete. So we have to look into the single molecules of these RNA, okay, and we don't have the technology is not there yet. And another thing is the living cells, the RNA, microRNA, and long non-coding RNA are known to have very low concentration and have a spatial and temporal dependencies, because these things are produced by various cells in various organs at various times, depending upon the progressing of the life cycle of these cells. So, to catch all the RNAs, very difficult. And living cells are very delicate and cannot withstand attack by our very crude instrumentation. So there's a lot of things to be done, but I think even with the very limited things we know now, I propose human beings are programmed in force. Thank you very much. Okay. Okay. Thank you. Lunchtime, right?
It is
Okay.
lunchtime.
Pass. Okay. So we'll go to lunch and see you back in 1:30, right?
What's up?
All right.
So have you gone about his robots?
No.
No? Okay, good. Wow.
Okay.